tutorial number 2. So now we're going to move on to cell processes. So basically I'm going to start with membranes. Okay, so last tutorial I talked about how the fact that membranes are semi-permeable. Okay, so that means that only some things can come in and only some things can come out, and it's the membrane that decides which comes in and which comes out. So basically the cell membrane is what we call a lipid bilayer. Okay, and it's made up of these things. So these are called phospholipids, and they're arranged in a line basically, and there's two parts to them. Okay, so you've got your hydrophilic heads and hydrophobic tails. Okay, so what do we know about the word phobic? Phobic means afraid of. That's how I remember it. So phobic, so basically the tails of these phospholipids, so let's write that down, phospholipid. Tails of these phospholipids are hydrophobic, so they don't like water. Okay, so basically the heads of the molecule likes water and the tails don't. So they basically form this double layer, phospholipid, and that's why we call it a bilayer. Now inside of this phospholipid layer, so some things can travel straight through the membrane. Okay, so remember the membrane is the thing that surrounds the cell. Okay, so some things can pass straight through, so things like some gases can just pass straight through. Okay, and we call that diffusion. Okay, so some molecules have no problem going across the membrane. So what we call diffusion, okay, so another word that is important here is osmosis. So diffusion basically is the movement of gases across the membrane, whereas as osmosis we're just talking about water. So both of these things will kind of diffuse across the membrane without much help. Okay, so they'll just diffuse straight away. In cells that need extra gases or extra water, so cells that have a lot of water passing in and out of them, so things like the cells of the bladder, cells in in your kidneys and things. So those cells need lots of water, so they have these transport proteins. And these are things that basically they kind of sit in the membrane. So if you just draw one there, they kind of just sit in the membrane and they provide kind of a channel through the membrane to allow these things to go through faster. So we call that an aquaporin. Okay, so if we drew like just a little diagram of what one would look like, so basically it'll just kind of be a channel, and then you've got your membrane. Yep. Okay, and then the water can go straight through and into the cell. Okay, so that occurs in something. So basically diffusion and osmosis are what we call passive transport. Okay, passive transport means no ATP is required, so no energy is needed. It just happens by itself. Okay, so how it happens is that the movement of the gases or the water or oxygen or carbon dioxide or whatever will always flow from an area of high concentration, okay, to an area of low concentration. So what do I mean by that? Lots of molecules here and only a few molecules here. So what's going to happen is life needs to be balanced. It wants to be balanced. So there's space over here for these ones. So what will happen is these ones will kind of casually kind of drift over there so that each side is equal. Okay, so you end up with less molecules here, more molecules here. Okay, it's just like what happens when you open some perfume in your room or whatever. You spray some perfume, and eventually the person on the other side of the room will smell the perfume, and that's just because the molecules have kind of, you know, spread out and moved over that way. Okay, so this happens also because particles are constantly in motion, and they're always wanting to kind of balance everything out. Okay, so another thing that I didn't mention, so these aquaporins here, these things, it's still diffusion, okay, but because it's being helped by this protein, we call it facilitated diffusion, and that just, or facilitated osmosis, that basically just means that it's being helped. Okay, so we've done a lot about osmosis, so you guys should be really kind of aware of osmosis. I'll just do a really super quick diagram of osmosis. Okay, so osmosis is the movement of water from a high concentration to a low concentration, so just like we were talking about up here. Now, if you've got, we did carrots or potatoes in different beakers. Okay, so we've got our carrot here, or our potato, or whatever. Alright, so you can see that. Alright, now the carrot naturally has, let's say it's a carrot. I'm going to colour it in this colour. Okay, so let's say the carrot. Carrots naturally have salts and sugars in them. Okay, so they're not pure water. Obviously, they've got they've got a taste, so they definitely have some kind of extra kind of sugars and things inside of them. So they've got a lot of starch and they've got some sugar. Okay, so basically, if we put the two carrots in different beakers of water, so here we've got pure water. Okay, and here we've got a salt solution or a sugar solution or whatever. Okay, so in this H2O one, you'll find that there's more sugars and things inside of the carrot than outside. Outside there's no sugars because it's pure water. So inside of the carrot you've got more sugars and more salts and things. Okay, in comparison to this one, if this is really super salty water, you've got lots of salts inside of here. Okay, you've still got a few salts inside of the carrot. Okay, but you got way more outside because this was really super salty water. Okay, so what's going to happen is, because water has to move from a high concentration to a low concentration, it wants to balance it out. Okay, so in this case with the water, what you're going to find is there's a high concentration of water outside of the cell than there is inside of the cell. Okay, comparatively, you've got a higher concentration of solutes or salts inside of the cell compared to outside. So what's going to happen is, just the same way as this, okay, you're going to have water molecules moving into the cell to try and balance it out. Okay, so basically it needs to balance it out. So the water is trying to dilute that salt in the carrot so that it equals. Okay, so in this situation, there's kind of almost no way that it would equal the same, because what will happen is the carrot will keep growing, and then it'll get to a point where it can't keep growing anymore. And there's no way you can make a carrot into the same water concentration as pure water. So basically what will happen is this will keep growing, you'll get a kind of swollen carrot, and then eventually if you leave it for too long, the cells are going to start bursting, okay, because this is hypotonic. I remember how I was talking about, I remember it, the hypo, the O, looks like something that's really swollen. Okay, comparatively in this one here, what we're going to have is there's more salt on the outside than the inside. Okay, there's more water on the inside than there is outside. So what's going to happen is the water's going to move out to try and balance it out. It's going to try and dilute that salty solution. The salts can't move. Okay, they're stuck in there. It's just the water that can move, and it's the water that wants to come and balance it out. So the water's going to rush out of the carrot, and what you're going to end up with is a little tiny shriveled carrot. Okay, so that'll shrivel to as small as it can get, and then can't get any smaller. Okay, so that's when the osmosis is going to stop. Now we call this a hypertonic solution, and remember how I told you guys, the E to me looks like it's all shriveled. Okay, it's a shriveled E. So that's how I remember it, hypertonic with a little shriveled E, and hypotonic with a big round O. Okay, so basically that's osmosis. It's trying to equal out, and if it did equal out, maybe if this was a little bit less salty, we will call that solution isotonic, and that means that the water is balanced on either side. Okay. Okay, so that was osmosis. Okay, so now I'm going to talk about active transport. Okay, so active transport happens when you have ATP. ATP stands for adenosine triphosphate. Okay, and it looks like this. You got an adenosine molecule. Tri means 3, so we have 3 phosphates attached to the adenosine. Okay, then what happens is, when it's triphosphate, it means that it's charged up. Okay, so this is energy. It's a very high energy molecule. Okay, when it is not charged up, we call it a DP. Okay, D means di, so it's adenosine diphosphate, which means it only has 2. So it's lost this one. This here, if I do it in yellow, it's got like high amounts of energy in that bond. So when that phosphate pops off, a whole bunch of energy is released, and that's what the cell will use. And then it'll go back to this one. So that's not charged up. So that needs to go back through respiration to get charged up, but we'll talk about that later. Okay, so active transport needs this extra energy to happen. Why? Because often active transport is going against the what is it? Concentration gradient. For example, if I've got a whole bunch of salt molecules on this side of the membrane, here's my membrane. This is my membrane. Okay, and then I've got only a couple salt molecules over there. Okay, so basically active transport would be, instead of the molecules going this way, as they would naturally do, I'm pumping these guys over here. Okay, so what I need is a pump. Okay, so it's basically a protein, and I have to pay a fee to use this pump, so I have to pay that in energy. So I have to pay ATP for this pump to work. Okay, so what it does is it gives the energy so that I can pump these across. So this is quite important, especially in salt regulation and things. So to get the salts across the membrane. Okay, so this is very important. This ATP in a lot of processes that we're going to talk about. Okay, so that is active transport. One more thing that we're going to cover just for transport across membranes is endocytosis and exocytosis. Okay, so you got your cell, here's the cell membrane, here's our nucleus, where's your nucleus. Okay, so basically endocytosis is what we call when particles are outside of the cell, and basically what the cell does is it kind of sends out these projections. Okay, and it engulfs these particles on the outside. Okay, and it kind of forms like a membrane around them, and it brings them into the cell. So you end up with kind of a vesicle with all of that stuff on the inside of the cell. Okay, so that's endocytosis. I don't know, that's how I remember it. Okay, exocytosis just means the opposite, so it's kind of the cell getting rid of stuff. So basically the vesicle will move to the side of the cell. It'll fuse with the cell, so you'll end up with, it kind of fused onto the side. Okay, and then it'll basically kind of just pinch off. Okay, so that is exocytosis. Okay, and then this one is endocytosis. This one's easy to remember. Exocytosis, exit. Stuff exiting the cell. Okay, a little differentiation for that. We've got this thing called penocytosis. And that is basically cell drinking. And it's the same as this, but instead of it being molecules, it's just water. So yeah, same with so basically this is endocytosis, but we've got cell drinking, and what we've also got is phagocytosis, which is cell eating, and that's this. Okay, they're both types of endocytosis. Sweet. Okay, so basically that is it for transport across the membrane. Then I'm going to go into a little bit of cell size and shape and start on photosynthesis and respiration. Alrighty.